Hi guys, I hope you're doing very well. This is our third capsule, and I think it will be a very interesting one because we will learn how to design primers. So as you know, our next practice is PCR, so I think it will be very useful to you if you know how to design and those primers. And we're going to we're going to work using an example. Uh, in a medical case, and I hope you enjoy it. So let's begin. So we have this case. So as you know, brain tumors is one of the most common causes of death, and there is type of tumor that is called meningioma. So the meningiomas are tumors that arise from this layer, from the arachnoid matter, because remember that to cover the brain we have many layers that protect all the nervous tissue. So one of these layers is one of the very thin layer. It's commonly to have like overgrowth and cell proliferation, so we have a meningioma. So usually it is very known that these kind of tumors are caused by mutations in those genes NF2, SMARCB1, SMARC1, and SUFU. All of these mutations have been previously reported and they have been related to disease susceptibility. But we have two cases, in this case the son and the mother, that they were screened for mutation in those genes because they have multiple meningiomas. So we know, or we assume that it can be genetic because the son is affected as well as the mother. So when the both were screened, interestingly, we discovered that there are no mutations in those genes. None of them have mutations in F2, SUFU. So it was quite strange because so we thought that there must be another gene that may be causing the disease. So we performed genomic sequencing and we found a genetic variant, RONX3. In both, the same variant was found in the son and in the mother. So once we found the variant, we required to screen or to sequence through Sanger sequencing to confirm that the mutation is really there. So what do we need to do for that? We need to design primers for the exon that we know that the variant is within that exon, so we can amplify the exon and see if the variant is really there. So let's go, and we're going to design the primers in the exon where the variant is supposed to be. So let's go to Ensemble. Okay, excellent. So for these cases, we're going to use the version 37 because remember that, as I told you, many hospitals and labs are still using this version because if they want to use the 38, they need to update all the bioinformatic pipelines and all the data sets, so it's going to be difficult. So they are still using the 37. So let's choose the 37. If you want to pause the video, and if you want also to do the activities as am I doing them, so I think it will be very useful to do that. So let's right here